Hello everyone and welcome back to the channel. Now, you probably saw or heard on the news the scientists, after decades of research, has finally managed to digitally map the complete brain of a fruit fly, which led to further insight into neurological disorders of humans. Well, today we're going to take a look into this new discovery and see for ourselves why this is important, how this was possible, and what this means for the scientific community. We're first going to start off with a question. Why the fruit fly? Why not forward flies, for instance? Well, the fruit fly, professionally known as the Drosophila melanogaster, has 140,000 neurons brain with more than 54.5 million synapses, which makes it a large but tractable target for neural circuit mapping. Now, the reason why they've emerged as a powerful model organism in genetic research is because of a number of reasons. First, the fruit flies with around 140,000 neurons are complex enough to exhibit behaviors like, for instance, learning, memory, or even sensory processing. But they are small enough to be fully mapped at a cellular and synaptic level. This makes them a very practical choice for high-resolution studies of neural circuitry that would otherwise be challenging for larger animals, perhaps mammalian animals like humans. But secondly, they share about 75% of the genes that cause diseases in humans, which means that they can allow for researchers to study how genetic mutations may lead to neurological disorders in humans and so on. Also, they're quick to reproduce. They have a lifespan around, up to 2 weeks, so a new generation is produced virtually every 10 days. This makes them very appropriate for a lab setting as well. But most importantly, they're very easy to genetically modify and study. Techniques like gene editing or transgenic approaches that introduce or remove certain genes, label neurons with fluorescent markers, and activate circuits are easy to apply to fruit flies. And this is a classic example of that, where somatic edits have been made to fruit flies. Okay, so we now know why such research has been conducted on fruit flies specifically. Then why is this research altogether so important? Well, the reason for that is because to conduct comprehensive neural circuit mapping, electron microscopy is needed. And the fruit fly brain was too big for the conventional EM to handle. But the new discovery allowed for a novel high-throughput EM platform called TEMGA2 in order to create a high-resolution complete electron microscopy volume of an adult Drosophila melanogaster, and it was equipped with an SCMOS camera for fast image capturing. And in the process, of previously unknown neuron type of MBCP2 in the mushroom body of the fruit fly brain associated with learning was discovered. This will be elaborated later on. Now, let's dive into the actual processes of how this was possible, but before that, let's look at a couple of key terms. First is connectome. It's a comprehensive map of neural connections in the brain, and we saw it in the first slide right here. This is an instance of a connectome. Next is the mushroom body. The mushroom body is a pair of structures in the brain of most invertebrates, and it's involved in the processing of sensory information, learning, and memory. And in the case of the fruit fly, it was right here. Right, and Kenyan cells. We won't probably speak of these cells in this video, but in the next video, but they're the neurons that make up the mushroom body. Okay, that's pretty much it for key terms. Now, let's move on to the actual processes itself. First stage is with specimen preparation. This is divided into 2 subsections. First is sample preparation. They started with the brain of an adult female fly, and the brain was fixed, and it was stained with heavy metals in order to enhance contrast. And then it was later embedded in resin in order to provide structural support for the delicate tissues. After that was the sectioning stage. Using an ultramicrotome, they sliced the brain into ultra-thin sections, which was around 40 nanometers each. And over 7,000 sections were created to span the entire brain depth. And this was around 250 micrometers, which allowed every detail of the brain to be captured layer by layer. Next is imaging with the TEMGA2. Now, a specialized high-throughput TEM platform was developed for this project, and this was called the TEMGA2. And the setup included high-speed SCMOS, a scientific complementary metal oxide semiconductor cameras, which allowed quick and precise movement of the sample to speed up the imaging. Now, the TEM is a type of microscope that uses a beam of electrons in order to create highly detailed images at a microscopic scale. And the reason why the development of the TEMCA2 was particularly important was because it compensated for the flaws of the conventional TEM and the challenges that it faced. Now let's recap the problems that we had. First problem was with brain size. The Drosophila brain volume made it hard to map because it required both high-resolution imaging to capture every single tiny synapse and a large-scale coverage of the entire brain expanse. And the typical transmission electron microscopes, the TEMs, operate very slowly and cover only small fields of view, which would make the full brain mapping process quite impractical and time-consuming. But the TEMCA2, firstly, it had parallel imaging with a camera array. The TEMCA2 uses an array of high-speed cameras that can capture multiple images simultaneously, which dramatically increases the throughput. And with each camera capturing its own small field, the TEMCA2 essentially tiles or stitches these together in order to create one large, high-resolution mosaics of each section of the brain. And it also had a very high speed of imaging. Each thin section of the brain can be imaged in less than 7 minutes with a data acquisition rate of around 50 million pixels per second. Now, this speed was approximately 5 times faster than the earlier conventional TEM systems, and it reduced the entire imaging time to a very feasible level. So basically, in summary, the TEMCA2 allowed for mapping of the fruit fly brain by being A, very fast, and 2, conducting parallel imaging with a camera array. Another development made was with automated imaging. They built an automated system, an automated transport and positioning system, or also known as the ATPS, that could swap out sample grids and image sections for extended unattended imaging sessions that wouldn't necessarily require human presence. Now, this reduced the time taken for their imaging sessions, which enabled them to scan each brain section in under 7 minutes. And with also the imaging collection process, with each section being scanned with TEM to create high-resolution images, this yielded an exceptionally large data set with around 21 million images in total, which totaled over 106 terabytes.